Why don't we sort of cut here for a moment? I think that would probably work better.
So in the evaluation of individual studies, it sounds like you've got two coders, two people who are independently looking at the studies. I take it they do so independently. And then you have some process for conflict resolution. And do you have a process for training the coders or doing intercoder reliability and getting what sufficient levels you think are reasonable? Can you just elaborate on that a little bit?
Yes, to both the questions. So for the process of evaluating the studies, yeah, we do have two individuals that will enter their documentation into HAWK, which basically that's the tool that we use. And at the end of the process, they'll meet again and go over their evaluations. And in the event that there's some disagreement that they can't reach consensus, we'll bring a third person to serve as a tiebreaker. But I don't think we've encountered that so far. So most of the time we encounter the same issues. It also becomes, at least for animal talks, as well as for EPI, very discipline specific. So when evaluating studies, there's a little bit of meeting ahead of getting started to discuss what is the focus going to be. In an animal toxicity study, you could get data on 5 different health domains, but you're really interested in only 2. So those are decisions that are made ahead of time before you get started.
Okay. And I'll just add that, part of the process during that discussion before we start study evaluation is developing the criteria that is exposure and outcome specific. If there's some uncertainty about it, if it's criteria that hasn't been used before, for example, there would be pilot testing to see that both are on the same page, and then before moving into full study evaluation. And we have developed some criteria that we can then use for that same outcome when we encounter it again. There was something else I was going to... the prompting and the follow-up questions that I showed on the epidemiology slide, for example, would be used to develop those criteria.
And what I wanted to add is, when I was at the National Toxicology Program, we were involved working with a collaboration with the developers of the Robbins Eye tool. And so as part of that, trying to think about how to extrapolate that exposure, we did this survey of trying to actually compare ratings based on sort of an approach that's very similar to EPA, OHAT, Newcastle Ottawa scale, and then having sort of a totally unstructured expert in the field assessment of the study. And it's not published yet, but sort of the preliminary findings our hypothesis was that because the underlying content was so similar, they were probably going to sort of rate them similarly. And I think we found correlations across those different tools and across raters being sort of in the 85% up. So it seemed like, in that case, what we deliberately wanted to interrogate is making sure that the raters had access to topic-specific experts. Because if you don't, then it can go wildly off the rails. But with that caveat, then we found that there was sort of a lot of, maybe more consistency than we expected.
Where in the process or there might there be availability for the public to look and disagree or agree with the evidence evaluation of the sort you're doing? Is there any place you plan to put that in or might put that in?
HAWK actually would serve as a platform for the audience to look into the evaluation of individual studies and see the rationale for the decisions that were made, whether it was high quality, low quality, et cetera. I guess that's something that we have to think about how that would be incorporated into like a paper version of the assessment, or if it's something that is more interactive, that the audience gets access to the HAWK database that gets generated and can scroll through, depending on their interest, the studies that they want to look into, the outcomes that they want to look into.
And I'm sorry, you had asked another question about across the coders and consistency that I don't think we've quite answered. And Chris, I'm surprised you didn't say it, because when Chris came, she said it's really important that we use a conserved way of filling in the different domains, how you answer it. You want to put, what your final conclusion is first, then, what the primary basis for your conclusion is and the rationale for that explanation. And so to have the coders have training on, we've had retreats to train on how to have very consistent ways of entering the information across coders, so we have a very robust way of comparing those reviews. So I just wanted to make sure we address that part of your question.
Karen?
So actually, just one quick clarification on that, and then a question. So in the IRIS assessments, is there access to the individual elements of the risk of bias for each study?
Yeah, you won't find the rater's name, but you will find the final rating, and then sort of be able to sort of see the rationale behind that rating. You'll be able to see it interactively as well as download it via Excel files.
Okay, so you won't just see the overall confidence, you'd see the elements.
Correct.
Okay.
Maybe one of you can describe how that hero will be transmitted with the
Yeah, I think we do some of this in session 3 when we talk about sort of the tools and use of Hawk.
Okay, and then
And it's also in the chloroprene. There was only one epi study, but there is sort of an interactive. It's a PDF, but you can click to sort of access the Hawk content.
Okay, so then my original question, could you talk a little bit more about reporting bias and how you assess that? I saw what I think was some differences across the animal studies and epi studies. So for example, in the animal studies, when you showed the table, it looked like it was being assessed in the same element as attrition bias. And then in the epi table that outlined the elements, I didn't actually see it there. So could you clarify what's being assessed in reporting bias?
While Xavier brings up the slide, I'll just say that for the most part, we consider reporting only as far as it affects our ability to assess bias and sensitivity. So reporting alone is less important than can we figure out the information that we need to determine whether there are bias or sensitivity issues. As far as I'll talk about epidemiology, and then I'm sure Xavier will chime in on animal toxicology. That's why it's not a separate domain for epidemiology, as we just consider it as part of our evaluation of other domains. So if there was, for example, not enough information reported about population selection, the study would be rated down for population selection unless we could get that information from the author or something like that.
And so for animal talks, perhaps some examples might be the best way to explain it. What we're trying to get to is that the question is, did the authors report on everything that they said they were going to measure in the study? Were there any outcomes that were left out? If each treatment group consisted of 10 animals, and you would expect to see that in the results, the sample size is 10. And if not, is there any kind of clarification of how animals were selected for one outcome or another? So those are the sort of things that we look into.
Follow up here. And Karen sort of got to a question I was going to ask you. You might go back to authors if a published report is incomplete, and how would you make the determination that you would need to do so?
If there's a study that would be informative, if we had additional information, then we will contact the authors and give them an opportunity to give us additional information.
We have always done that.
Yeah, that's not new.
Yeah, and in particular, I think if we determine that for the purposes of analyzing the data set for consistency, if we can do something like a forest plot showing variation, and the central estimate, then, and that's not reported in a paper, we'd go back to them and see if we could get that kind of thing.
George, you laid down here.
My questions are answered.
Okay. Ilya?
So in evaluating animal and human studies, you have multiple domains. Do you have specific measurements on each domain where you apply these measurements consistently? So for example, you talk about human study sensitivity. You said, for example, we could consider sample size. So my question is that, do you have a set of fixed measurements on each domain by which you assess the quality of the study within that domain?
So you would develop criteria in advance of doing the evaluation that would explain what you would consider a good study or an adequate or a poor or a critically deficient study. And that would give you, we don't do any scoring where, if you tally up, the information and come to a final conclusion about how good the study is, we use expert judgment to evaluate the domains. I don't know if that quite answered your question, though.
Well, so I guess let me try a different way. So when you say sensitivity of this human study, well, you said that in your presentation, you said, for example, if the sample size is too small, maybe the study is not sensitive enough to detect that's one criteria. To me, that's one measurement. So how many different measurements you might consider on that particular domain? Do you have a fixed set of criteria or measurements for that domain?
No, there's not a fixed set of criteria or measurements for each domain. So we would start with these prompting and follow-up questions to develop criteria. So some outcomes might have a lot of detail, some might have only a little bit of detail, and similarly for the other domains.
Okay, well, to me that's kind of interesting. So you don't have a fixed set of criteria for each domain. So your evaluation becomes sort of the criteria becomes case-to-case situation.
Right, because it has to be exposure and outcome specific.
Okay. Now, once you have, let's say, in a particular domain, you do have a number of criteria that you adopt for that particular study. How do you come up with an overall ranking or evaluation for that domain as low, medium, high in quality? Do you have a fixed algorithm to weight different criteria, or how do you do that? I mean, if you do, whatever you do, is this well documented in your protocol or manual? So
there's not a fixed set of domain ratings that would then lead to your overall rating. So here are the definitions that Xavier went through for your overall confidence in the study. And the reason that we don't just say if you have this number of goods, you get to high, is because the limitation may be more severe or less severe, and we want to be able to weight in terms of the impact on the effect estimate, and we want to be able to weight those when we're coming to our overall confidence.
Thank you for your explanation. My concern is that in this case, how do we convince a person like me there is a consistency in this process?
I would, and I'm not sure if this answers or not, but when you had your slide up with the prompting questions, so those prompting questions are going to be pretty consistent across assessments. The responses to those are going to be sort of tailored to the exposure, the outcome. So there is consistency, I think, in the prompting or signaling questions. And one of the things that I think we've struggled with, and when you think about maybe some of the other bias tools, and this sort of gets at, an algorithm to get there, is I think when the Robbins I, there's sort of the overall can't be any higher than the lowest individual domain. But you might have things like adherence to the protocol as a domain. But here, I think in our field, one of the things we struggled with is we're really concerned about sort of the exposure assessment, the sensitivity of the study. And so that's where it's hard to sort of maybe some of this weighing, it's hard to sort of be prescriptive X, Y, and Z. But I think that there are certain domains, especially exposure, sensitivity, that if you had issues there, then it would be hard to consider the study to be high quality.
Just to follow up on that, let's say you have a series of insensitive studies where you don't expect to find anything, but then you do, and it actually then would translate to high confidence. All other things being fairly well. So, there's this negative versus positive bias. And so how do you deal with that? Is that done at a sort of general level, or? how you deal with the issue where you actually see something where you have a series of things that biases towards the null. And then overall that might even feed into a high confidence finding.
Well, I hope this answers the question, but for animal tox studies and EPI as well, we try not to look at the outcome or the results of the study if there was a signal that was reported or no response. What we're trying to focus is in the methods of the study and that particular outcome that we're evaluating. So like I'll pick up the example of phthalates that you'll see in the posters later on. There we're interested in hormone measures and effects that are downstream from those changes in hormones. So we want to see the studies to be designed in a certain way. If it's going to be a gestational exposure study, does it expose during the critical window? What are the outcomes that are looked at? Are those things that are considered established? How was it measured? And all of those play into criteria, if you will, as to how you go in to judge the study, but we're not looking at the results at that point.
I guess I would add to that, though, but that's only for the individual evaluations. And like Xavier mentioned, one thing that I think is very important is that we carry through the rationale for that overall decision. So if the rationale for that low confidence rating was that it was a bias towards the null for insensitivity, when we're reviewing the results and extracting the results and doing our synthesis, obviously we wouldn't treat that study as a low confidence study at that point. We're always keeping the rationale for that decision and not just the medium, high, low confidence, because that rationale is very important to the decision process during synthesis.
A quick follow-up clarification. So are these ratings applied on a study-by-study basis, this slide, or is it for the general level, the overall across a domain?
Actually, they're applied. They could be study by study if you're just only interested in one outcome in the study, in the case of animal studies. But if you're evaluating, let's say, liver and neural effects, you may see that the study is really well designed for one outcome, but not for another. So for one study, you could have two different overall ratings, depending on the outcome that you're evaluating.
Let's see. Okay, I have two questions, I think. Let me get mine in, and then and answer quickly, because I think we've got stuff to move on to. So question one, answer yes or no. Do you consider these tools final? Yeah, are they in their final form now? Or are you still working on them? Yes or no?
Yes.
I think the questions are stable. I think we probably, some of this calibration of, how do you sort of get to the point of coming up with the overall depending on sort of the domain patterns. I think we have some internal calibration to do, but I would say on anything that goes live, we are being super obsessive on calibrating on those.
Okay, yes.
Well, but yeah.
All right, next question. Second question, I haven't heard any mention of sort of generalizability or external validity as you review the either tox or the epidemiology. And so where are you tracking that? And that sort of is certainly, I think, an issue in extending either the results from a particular species for a particular outcome, or certainly with epidemiology.
It can happen at the pico stage, especially in cases where you know something is not going to be generalizable, then you might not consider it relevant. But it's also going to be covered in the evidence synthesis and integration session.
This was, I think, just a minor point. I think, Javier, you had mentioned about reaching out to colleagues outside the EPA. So I was just wondering how you decide when to do that and who you reach out to, and if there's any kind of formalized process for this.
My experience has been that it's sort of informal as you dive into the database for the chemical that you're looking at. There are several tools and sources of information out there, like description of guidelines for studies or recommendations for, say, histopathology for reproductive effects. And I keep saying repro because that's the focus that I have in IRIS. But But whenever something comes up that I don't understand, what I'll try to do is reach out to the study authors or some of our colleagues inside EPA to ask things about, for instance, is it better to take a hormone measure in the tissue that's producing the hormone or serum hormone levels, and what are the pros and cons of each, and try to get some of that feedback when there's an area that I encounter that I don't know.
But we have gone outside. We've, in one example for an assessment where we were evaluating asthma studies, we, through a contract mechanism, recruited a number of experts in asthma epidemiology to help us to understand the differences in the validity of various outcome ascertainment methods that were used in different epidemiology studies. So.
Okay, I think we'd better go on. Barbara, I think you've got a lot of important material to present.